Prokaryotic replication fork. DNA replication involves many different enzymes to complete the process and occurs at the replication fork. Every time replication occurs, there's a leading strand and a lagging strand. In this diagram, the leading strand is on top. We can determine this as continuous replication is taking place on this strand going from 3' to 5' towards the replication fork. Nucleotides are only added to the 3' end of DNA, leaving replication to occur in the direction of the fork continuously as replication occurs from 5' to 3' on the complementary strand being made. That leaves the bottom strand as the lagging strand. It's considered the lagging strand as discontinuous replication occurs, making Okazaki fragments, as replication only occurs in small pieces. The lagging strand goes from 5' to 3' towards the replication fork, as the two strands are antiparallel, making replication occur in the opposite direction of the replication fork. In prokaryotes, there's only one origin of replication, also known as where replication begins. Gyres can be found relieving tension between the DNA strands to prepare it for helicase to come through. Once gyrus relieves the tension, helicase follows behind, unzipping the DNA strands by breaking the hydrogen bonds between the bases. Although the hydrogen bonds are breaking, the phosphodiester bonds remain intact. This is because these bonds form the sugar phosphate backbone between the nucleotides, which stay bonded to one another. Once helicase unzips the DNA, it is crucial to keep the two strands separated for replication to occur. This is the job of the single-stranded binding proteins, as they make sure the strands stay apart. Now that the DNA has been separated into both the leading and lagging strands, specific enzymes can come to help form two identical pieces of DNA. The first important enzyme is RNA primase. This enzyme plays a crucial role in letting DNA polymerase III know where to bind to start replication. It lays down RNA nucleotides telling DNA polymerase III to start here, and this is called the primer. Once the primer has been made, DNA polymerase III can come in. The job of this enzyme is to add DNA nucleotides that are the complement to the pre-existing DNA strand to make a complete piece of DNA that is just like the original. This enzyme knows what to add, as adenine will pair with thymine and guanine will pair with cytosine. Since DNA polymerase III is adding DNA bases, it is important that we replace the RNA primer with DNA bases to create a complete piece of DNA. This is done by DNA polymerase I, who replaces all of the RNA with DNA. As we are nearing the end of replication, there's only one more step. This step is completed by ligase. The job of ligase is to act like the glue and combine the Okazaki fragments on the lagging strand. Once everything is glued together, we will now have two identical pieces of DNA from the process of DNA replication.